Okay, so this would have been your PAG, dissecting vascular tissue in a plant. So we would have got some celery and we would have sectioned it and stained it and looked at it. And I'm going to just talk you through it because you will be doing it when you come back to school. And what I might do is get you to do a drawing. Anyway, let's have a look at this PowerPoint. Right, so you would have dissected a stem and then stained it, and you were going to be doing low power. Now when it says high power, you were only going to go up to the medium, which can I remember which one it is, times 100. You wouldn't have gone up to times 400. You can't see a thing on that one. So a low power plan and then a little bit more detail. So I'll take you through some of those. So first of all, you need to remember how to do scientific drawings. Now a low power plan is just the bare bones. It would just be like if I was drawing one of the labs, you would just do where the doors are, the windows are, and the benches. And then a high power detail, you would take perhaps a bit of the front bench and you would draw the people and the chairs and the bags and the coats and the pencil cases and all of that kind of stuff. So you'd have much more detail. Now hopefully you can spot the error on this particular diagram. It's okay, it's a good diagram, apart from all of these are on one side, which means they're a bit squashed in, which means you can't do horizontal lines. I don't have a ruler, okay? Forgive me my non-horizontal lines. So you would use a ruler, they have used a ruler, but some of these, say maybe every second one, you would move to the other side so you can spread it out much more, and you want nice ruled straight lines. Okay, so this is a high power diagram. Again, if they had spread some of this out onto both sides, if they put their diagram in the middle, then they would have been able to spread that out a little bit more. But you can see these aren't just labels, these are annotations. It's giving you detail. Now detail can be different things. It can be function. It can be, I couldn't remember the word for colour. It can be colour. It can be something descriptive to allow you to identify it. Okay, now the other good thing that they've done is they've added a scale bar here. But you can see they haven't drawn every cell. They have drawn a representation of what they could see. Okay? Right, so it's fairly obvious to see this one, the lines aren't going out to the side, they're not using a ruler, these labels are all over the show, they should be nice and straight. Your label lines should be in pencil. You can write the words in pen if you want to, but you can do those in pencil. These ones are better, but again, why didn't they just do that one straight? Why didn't they spread these out so that they could do them straight? You can always, if you need to, if you don't want to kind of get in the way, you can always go up and out, down and out. That's a much better way of doing it. Or if you've got two things very close together, you can come in and then split it like that. But they should be straight and horizontal. Okay? Right, so this is what you would have, you wouldn't have seen anything quite as good as this. So this is what you get if you stain with toluidine blue. Now I'm going to try and highlight things. So that would be the xylem, all of this stuff here. The phloem is that layer there, so that bit there, and then the sclerenchyma is that bit there, so kind of that band. Okay, so let's just get rid of that. So yes, I mean that is a very, very nice diagram, and so all of this stuff here would just be parenchyma, it's just packing tissue. Okay, so this is what more like what you would have seen. So you can see here is the xylem. That's really nice and obvious. The xylem is really easy to spot. It's the easiest thing to see. This would be the parenchyma, just the packing tissue. Now this is more likely to be sclerenchyma, and so that layer in there is probably the phloem. It's difficult to tell on this because obviously You're not looking at the real slide. What you would do when you were looking at this is just do your focus up and down a little bit, just so you can get slightly better focus. Now you can see there's more than one layer of cells here. So it is quite a thin section, but it's not a perfectly thin section. So it's not just one cell thick, which is why it's a little bit more difficult. And so these were some of the others. So you can see on low power here, you've got the xylem there. That would be the sclerenchyma. It's difficult to see the phloem. You can see the xylem there very, very nicely. Depending on the colour, that is probably sclerenchyma, and that is probably in there, is probably the phloem. Maybe a little bit of this round here is phloem. It's difficult to tell. Yeah, I'd have to see it on the actual slide. But that's the kind of thing that you're going to get. And here you can see some collenchyma. It forms like little bean shapes, kidney bean shapes. And it's the stringy stuff that makes celery really kind of tough to chew and eat. And so sometimes if you cut through celery and you don't cut through it properly, you can pull strings off the outside, and it's that collenchyma that you are pulling strings of. Okay, so this is a slightly nicer one. You won't be surprised to know that I made these sections. So you can see the xylem there, you can see the phloem, that would be the sclerenchyma. Now this is a longitudinal section, so if you've got your celery going like that, a longitudinal section will be cutting down that way, and then you cut another slice very, very finely and thinly next to it. And what you can see here, what I really like about this view, is that you can see these spirals of lignin, and every now and then sometimes you can see it kind of ping out. It kind of looks a little bit like a bed spring that's kind of gone, and that's really cool. And that lignin, that spiral of lignin, is what gives that xylem its support, or its support ability. And so that dark green ring around each of these is this lignin that's stained.